Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com webinar titled Optimize the Value of Your Research Investment, Outsourcing Molecule Design. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host for this event. This webinar has been sponsored by Crescent Discovery, so a big thanks to them for helping to make this webinar possible. We're very fortunate to have Dr. Martin Slater here with us today, who's the Director of Discovery at Crescent, who will discuss some common challenges in molecule design and how they can be overcome with computational chemistry methods. And so without any further ado, I'm very pleased to welcome Dr. Martin Slater. Martin, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thanks, Liam. Thanks for the introduction. I'm going to start off with an overview of the talk. So first we're going to start off with, I'll move the question view out of my way a second, the value of computational chemistry in drug discovery. So what the challenges are, excuse me, and how to navigate through roadblocks to project success. Then we'll move on to the advantage of outsourcing molecular modelling, access to brains in our organisation. And then we're going to move on to describing a few case examples, how outsourcing has delivered value to our customers. And then we're just going to finish off with the actual organisation, Crescent, and how you could work with us. Okay. So, challenges in drug discovery. This is quite a complicated slide. It shows the interface, which is mainly between biology and chemistry, which is, medicines are usually developed to solve problems, disease areas. That's associated with a whole raft of biology. And what is interesting to us is how that interfaces with the chemistry. So it's a complicated, multidisciplinary, multi-parameter problem. And the main challenge is understanding enough about that disease and the data around the disease to be able to come up with a chemistry solution to that problem. So we're mainly looking for small molecule interventions in some sort of process. So that's the first challenge. The challenge for drug discovery organisations is getting their heads around all that information and data that they have to put together in order to solve those problems, but also navigating this sort of classical medicinal chemistry linear path, which is a little simpler. It's a cut down version of the previous slide. And this will be recognisable to most people that are involved in drug discovery. It involves target ID and validation, hit discovery, and then you move through this sort of linear path through to candidate selection. So one of the problems is a lot of the low-hanging fruit in this process is gone. All the easy targets are gone, and what you're left with are the quite difficult and challenging targets. There are also lots of different paradigms, so this isn't the be-all and end-all to that process. So in another context, the regulatory requirements for achieving drugs that are safe to be marketed is very, very challenging and becoming more and more challenging. So companies are trying whatever they can to reduce the risk of this process and make efficiencies. It's also very, very critical for the companies to efficiently convert their target portfolios into viable, developable drug projects. So how do we help with this problem? How do we get you from A to B in a more efficient manner? And that's really what the talk is about. And the way to think about it is, if you're making a journey, say we're going from Cambridge to Edinburgh, you could definitely walk. That's sort of a low-tech version of getting from A to B. You could get on a bike. You could also drive a car. You could drive a Tesla up to Edinburgh. You could go up in style and with a bit of technology. You could even fly. So you can use technology to help you get to where you want to get to more efficiently. And we can think about that in terms of this process. I've skipped a slide, excuse me. Just one slide. So this is a diagram of that same path going from target ID to lead optimization. And what I've added to this is a set of activities that basically augment that process, make it more efficient. And these are computational chemistry workflows and tasks that can basically help you on your way to achieving that endpoint more efficiently, and adding a technological solution to that process of getting from A to B. So deploying computational chemistry on a project can maximize the return on investment of that, can reduce R&D spend, and that's the main point of this slide. So we want to get efficiency gains in this process. So why outsource? So one thing is actually doing computational chemistry. So that's one thing. You can do computational chemistry if you have a computational chemistry group in your establishment, or you may not. So I'm going to talk about a couple of options. So if you are a big pharmaceutical company or a large organization, a research institute, for instance, you might already have in-house computational chemistry. You may have modeling, you may have IT support, infrastructure, you probably will have. However, in big pharma companies, generally these resources are fairly stretched. And actually over the last, I guess, 2 decades, those sort of R&D activities within big pharma organizations have suffered a little bit of a lack of investment. The size of the groups have reduced, but the size of the portfolios that they need to work on hasn't shrunk. So you get this problem where there's a capacity issue within big pharma companies and other organizations where they just don't have the capacity to deal with the large number of projects they're trying to prosecute, but also some capabilities have been degraded with time. Excuse me. One of those capabilities, for instance, is high throughput virtual screening. So we can help with that. We can host those types of capabilities and help that process move, even for big organizations. The other extreme is a smaller organization, excuse me, where maybe it's a virtual company. It's got a few board of directors, a head of chemistry, and you outsource everything. So you may outsource chemistry, and some of the biology maybe, outsource procurement certainly, and you may not be able to afford a computational chemistry team. So again, we can help with that rather than trying to find good computational chemists and setting up your computational chemistry group, sourcing software. It would be much easier just to outsource that, just as you would outsource medicinal chemistry to maybe India or China. There are excellent chemists in those places, and you don't have to hire chemists. So in the same sense, you don't have to hire computational chemistry, you can basically outsource that. So those are the arguments. So what we want to do is make this path a lot more linear, less bumps in the road, more efficient to get from A to B, and accelerate the creation of your commercial assets, reducing FTE rates, reducing the need to employ people and keep employing them, no need to get infrastructure, big CPU farms for doing big computations. We can do all that sort of thing for you. So this is an example of a return on investment. So one of the things that we do quite a lot is helping to design molecules. So, you can drive synthesis design using traditional standard medicinal chemistry rationale. But if you can apply these in silico methods, we can advise on, what molecules not to make. Don't waste your time making those molecules. They may have looked really good in 2 dimensions. They don't look good in 3 dimensions. They don't fit a hypothesis of how those ligands are interacting and how their activity is working through their structures. So that's a really useful thing to be able to do. We can also reduce the amount of wet chemistry you do by removing things that you don't need to make. Another benefit of using those is if you do use your internal chemistry, it's quite hard to find computational chemists. So there is a shortage of computational chemists out there. And now to the in silico advantage. So if you do the high throughput screening route to discover hit molecules, that's likely to cost you something in the region of $1 million, $2 million, whatever it costs, and it will be over a period of time, 18 months. The process is kind of random. You're screening a large number of molecules, and then you find hits through biological assays, and then you've got to work those up and try and understand how those molecules interact. So you can start this process with an in silico method. You're much closer to the 3D pharmacophore of what the molecules are doing at the molecular target. It's more efficient in that sense. It's also cheaper to employ in silico methods. And so you get this shrinking of the cost to do the same type of activity. We can do it quicker as well. So you get both a cost advantage and a time advantage in applying an in silico method. Just really makes sense to do that. The benefits of using a dedicated team who are experts in computational chemistry, that's fairly obvious. We can also help with deploying appropriate in silico techniques. So part of the problem is actually knowing which technique to use. So, we can help you work out what's a sensible strategy going forwards and then deploy that. We have powerful science. We've got modeling software and infrastructure. So we've got a big computational cluster for doing big calculations like molecular dynamics, FEP, library enumeration type things that require a bit more effort. Another advantage is resource on demand. So you can use those when you need us rather than, buy a computational chemist and then you've got to pay them, all the time, unfortunately. And then obviously we can work with you. We can make that a collaborative thing. It's backwards and forwards rather than throwing an action bundle over the fence and letting another organization just deal with that. So it's very seamless communication between the clients and ourselves. That's obvious again. Another facet of the advantage of outsourcing to the Crescent as an organization, we are able to help these organizations in medicines discovery or molecular design discovery in a variety of different ways. So that linear path I mentioned before, so the target identification, there are things that we can do there as an in silico method. The main focus for us is sort of the lead generation and optimization part of the process. So hit finding with virtual screening, QSAR once you've got hits, and SAR and de novo design for moving forwards to try and optimize those ideas. We also can be involved in the preclinical development side, the optimization of properties of molecules as well. So, through the pathway, but mainly this central part is the thing that we're mainly involved with. The other thing that is a key component of what we do is Crescent science. So Crescent science underpins the whole business. So we have a software part of the business and a discovery part of the business. The software there is focused on providing very accurate electrostatics around molecules. So we have molecular mechanics force field, gives you really good descriptions of the electrostatics. It allows you to compare molecules with each other very, very accurately. We can also use the same technology to describe the interaction between ligands and the protein targets. So the bottom picture there shows a protein pocket, and the electrostatics, cyan and red, around the pocket that match to the ligand's electrostatics. So that's something that underpins all the science at Crescent. But we don't just use Crescent science to solve problems. We'll use whatever tools are appropriate to solve that problem. In terms of the types of things that we do a lot of virtual screening. But again, it's not the only thing we do. We can do homology model building, QSAR, library design, a whole variety of things that you might want to do. And again, we apply these workflows appropriately to whatever the problem is we're trying to solve. So it's not, we've got a hammer and that's the tool that we're going to use, whatever the thing is, whether it's a nut or a marshmallow. We'll apply the tools appropriately. The other advantage is the expertise within the company. So we have expertise from the pharmaceutical industry, biotech. We've also got agrochem people. We also work with flavour and fragrance companies and the chemical industry. And also we work with academia as well. So there's a whole range of types of experience within the organisation. In terms of the team architecture, it is a team effort, so you're not contracting a lone modeler or a consultant. This is a team, so there's a whole support network around the in silico tasks that we're performing on projects. Okay, so I'm going to move on to a couple of case studies now. This first one is a project we've been involved with relatively recently. So this was with the University of Cambridge Department of Genetics. What they found through their academic research was a very, very interesting protein that essentially controls a component of signaling within bacteria. This signaling essentially controls whether the bacteria go into this sort of resting state where they're not metabolizing, they're just sitting there. The problem with those bacteria is they just sit there. You can't kill them with normal antibiotics. So it's a real problem, and the University of Cambridge were really interested in this in terms of developing novel therapies for urinary tract infections. So one of the problems with that area is persistence of infections. You get persistence from, as I said, the bacteria going to sleep or the bacteria forming biofilms. So this enzyme that they identified is a target which if you hit it, you can stop them going into this sleep state and stop the formation of biofilms. So it's a really interesting target. It's an enzyme, so we were able to have a look at this enzyme and define a geometrical and also a pocket-based approach to preventing this enzyme from working. We devised a virtual screening paradigm where we could screen a number of compounds. Obviously, the University of Cambridge, they weren't running on a very large grant at that time. They had a small amount of cash, so this was a very, very cost-effective way of trying to find active molecules from their target. So we did a few virtual screens. We've screened 6 million compounds. We identified some potentially interesting molecules. They went away and tested some of those, bought them first, and then tested some of them, a very small number, and actually got some really nice hits from that. That process, just getting like a proof of concept, the hits from that process, allowed them to then get a bigger grant to go back and buy a few more of the compounds that they couldn't buy in the first round and repeat that process. And they basically cycled through and ended up with about 30 hits from that process. And then they've gone into developing those molecules. Part of their assay setup was a cellular assay, so they didn't want to just hit the protein target with molecules. They wanted to hit it. They wanted to hit it in the cells. So there were inhibitors of the enzyme that were around, but they didn't work in cells. They couldn't get through membranes. So their assay that they set up, and what the virtual screen provided, was compounds which they would put through the cellular assays and get an output. So they were very successful. They've found molecules that are actually active at preventing biofilms. So they're sitting on some really nice assets, which they got to in a very, very short period of time through not a huge amount of investment of money. So it's a really, really nice example. It was really good to work with them. This is another example where we worked with an antifungal company. So this is this is published, an extract from a PNAS paper. So they had an antifungal target. We essentially helped them to understand their SAR. So they actually arrived at molecules via a high throughput screening method. So they had hits. They developed those by standard medicinal chemistry to a point. And then they really needed help to get much further forward in that process. So part of what we did with them was to try and understand the ligands, the SAR data around those molecules. They were really struggling to get crystallography data. This is it's a protein which the human version has crystallography, but the fungal species protein is actually, it's membrane bound. It's partly membrane bound. So once you take that membrane binding component out, you can't actually crystallize the protein. So throughout the whole of the project, they could not get crystal structures. So the only way to allow them to do rational design was to build a homology model. So this project was really focused on understanding their SAR and then building a homology model and getting an idea of whereabouts in the protein those molecules are interacting. So you can go and have a look at that PNAS paper and see what we did there. They have actually produced a first-in-class set of antifungal molecules from this effort. We didn't find the original compounds, but we did help them along the way. And it was, again, it's a really nice collaboration with them. Here's another example, completely different from the other two examples. So this was with a US firm. They were interested in describing fragment chemistry space. Our technology, which I already mentioned, which involves using electrostatics to allow you to describe and compare molecules with each other or fragments of molecules with each other, it allowed us to help them select the most diverse set of three-dimensional small fragments that they could then use to build a platform. So this company has built up. They've actually gone away and made, synthesized, and also acquired compounds, then built up this library of really diverse molecules that basically are a drug discovery engine, a fragment discovery engine. So that was, again, another really nice collaboration. And changing tack again, this was another project with a joint academic, a three-way relationship between an academic group and Crescent Discovery and a third-party CRO who did the chemistry. So we basically worked in collaboration with both the host of the project, the academic client, and worked very closely with the chemistry provider. This was a really challenging target. Again, it was a protease. It was a serine protease, which has been the death of a lot of activity in the pharmaceutical industry. Really, really quite challenging to get selective and active molecules in that area. We ran a virtual screen, found some really interesting molecules. The chemistry provider followed a slightly different route and found another molecule that was an interactor of the S1 pocket. Again, they got to the point where they were struggling with the SAR and to understand what was happening with the SAR. So this is an example where one part of the molecule was interacting with part of the protein. And again, what was really quite important were the electrostatic signatures of the molecules. You can kind of see that in the picture, but I don't want to spend too much just time describing exactly what that is. But it's essentially an electrostatic clash between part of the protein and the molecules they were building. So they needed to retune their molecules to try and make that interaction the best that they could get. And we helped them with that and understand their data. So I think this is the final example. This is a really nice one. We worked with an agrochemical company this time. They had a molecule that killed nematodes, and they wanted to develop that molecule and find another example molecule that killed nematodes and would be more suitable for an agrochemical formulation. We helped them find a new molecule. As you can see, the molecule is fairly simple, quite compact and small. We helped them find an alternative structure for that molecule, which ended up being a product for them. So this is a product that's on the market called Teoxazifen. The original collaborator was actually bought by a big agrochemical company called Monsanto, that you might have heard of, and this basically was one of their products. So it's a nematode killing molecule. Okay, so that's it for the case studies. Just to give you a flavour of how and what we do with our clients. I'm going to talk a little bit about working with Crescent Discovery. This is the sort of thing that our clients have said they have enjoyed. So the expertise, the speed and the flexibility, working with a team, not just a single expert. We have a global footprint. So we work with US, Europe, all over the place. I've said that again, I wasn't supposed to say that. Next slide. Another way in which we work collaboratively and seamlessly with our clients is by sharing that information in a way that's useful to the clients, the customers. You can send information by email. That's not recommended. They can be intercepted. We prefer to share information via some protected method. So we'll use SharePoint or Ignite for sharing files and information that are deliverables. Another way of doing that is interacting with a platform that we've built called Talks in collaboration with Elixir. That allows us to actually do live interactions with clients, sharing designs and data, and manipulating molecules in the context of maybe a protein live. That is a platform that can be distributed by the customer. They have the license. They can then share that information with third parties. So as I described in the case studies, if you're working with a client, the core client, and then they've got third parties that they're working with, they can disseminate information that way through talks. So that's a really nice technology solution for sharing information and data. There are a couple of testimonials for you to read. One of those, the collaboration described in the case study, was with F2G, which is an antifungal company up in Manchester. So that was a comment from Graham. Leave that up just a second, I can have a glass of water. Excuse me. So hopefully I've managed to convince you that using Crescent Discovery is a good way of making this process much more efficient, this linear process much more smooth, accelerate, again, the creation of your commercial assets. Finally, a little bit about Crescent. So we do have a proven track record of delivering CompChem solutions. We've provided over 300 projects, more than 300 projects for clients over the last 20 years. So we were founded in 2002, so we've been around quite a while. We're going to be around for a long time in the future as well. The software part of the company works with 15 out of the top 20 pharmaceutical companies. We're also successfully growing. We've got 70 plus staff across 3 continents. So, you know, you're in safe hands with Crescent Discovery. So I think that's my last slide. So if you want more information about Crescent, obviously you can get that through scientist.com. You can also subscribe to Computational Chemistry News from Crescent. I've got a great website if you want to have a look at that. And then I think that's it. So thank you very much for your attention. And we're open to questions.
Fantastic. Well, thank you, Martin, for a really great presentation. Yeah, we will be taking some questions now. So let's jump right in here. Could one also outsource medicinal chemistry to Crescent in addition to computational chemists? And if not, how do both MedChem FTEs in-house or outsourced to another CRO efficiently collaborate with the computational chemists at Crescent?
That's a good question. So we mainly focus on in silico part of the process. As I said in the case study example, we do and we have worked with a number of different CROs that are offering the chemistry part of that process. Drug discovery, as I said in the first couple of slides, it's got biology, chemistry, computational chemistry, ADMET at the back end, and before you go into the sort of the early clinical trials of molecules. We focus on the in silico part. We do have preferred partners for chemistry, so people that we've worked with and are happy to work with. But we don't do any of the chemistry ourselves.
Fantastic. All right, next question here. Do you think your services are seen as being competitive with internal computational chemistry teams?
Yeah, it's a good question. You would think so. However, just from experience working with big pharma companies and even small companies, we still work with computational chemistry groups in other organizations. Quite often there may be a lone computational chemist in a small biotech organization, so one person. And I think that's a situation where it's about capacity to do the amount of work that is required to be done by these small organizations. But also we have the heavy lifting capability that a lone computational chemist in an organization wouldn't have. So that's a benefit. In the bigger organizations where you have a computational chemist, or a group of computational chemists, again, it's about either providing backup for those chemists, those groups charged with trying to tackle a large number of projects, or again, it's maybe some of the heavy lifting that they either don't have or have degraded capacity to do. One of the things that we're doing, we do a lot of, is virtual screening. Ligand-centric virtual screening requires a lot of computation. If you don't have that a large maintained cluster with IT support around that, it's quite difficult and expensive to do that if you're not doing a lot of it. So that's one way in which where it's used by the big pharma companies that already have computational chemists or computational chemistry groups. I think we can complement pharma and organizations that have computational chemistry and provide an additional opinion. We have a team that's the size of a big pharma company's computational chemistry team now, so we have got that bandwidth and the expertise within it. We also have the advantage of working with a lot of different companies and getting exposure to a lot of different challenging targets. So we can help come up with ideas that maybe the big pharma guys just don't have time to think about.
Yeah, that's a really good point. Yeah. All right. So, good question here. How exactly are the results and the deliverables shared between you and the customer? I think you mentioned SharePoint a bit before, but can you clarify a bit about that?
Yeah. So as we work with clients, we write a fairly detailed proposal that outlines what the plan is. Part of that plan is what the deliverables are. So the deliverables might be presentation material, there might be protein structure files, chemistry structure files, SD files. You don't want to be sending those by email, as I said, because for two reasons. One is capacity of email, and also you don't want any of that material to get intercepted. So we use sharing ports. Some of our clients use things like Box or Ignite is very popular for US clients. We also use SharePoint, or we have in the past. So that's a secure way of sharing either documents, so Word documents, presentations, but also the results files, so protein structure files, ligand structure files. And it's up there in a secure way, and then the clients can get access to that and upload material for us to have a look at as well. So it's a two-way sharing. So that's one way of sharing information. We also share information by live demo. So if we do a project, then we may describe the output of that project, not just by a bundle of files for them to have a look at and try and work out what's going on. We'll present the results in a presentation format, but also in a live modeling session where the client can see the 3D modeling from our software or and interact. So that's one thing we do very regularly is have interactive meetings with our clients at discrete intervals. Another way, and again I already described this, is the Talks platform. It's a very innovative way of sharing information that can be done to demonstrate live modeling, and that's two-way. So the client can basically rotate the molecules in the context of a protein and say, oh, okay, I see what you mean about how that molecule maybe doesn't fit, or what the interactions are. And you know, we can drive that, and we can let the client drive that. That's a really fantastic way of sharing information. Again, very efficient. Rather than writing down, you know, reports and building presentation material, you can actually get right into the nitty-gritty of the project. You know, computational chemistry, the molecular interactions through that platform. So that's really good.
Yeah, that's neat. That sounds like a really interesting way to share your results. And I think it leads in nicely to a next question here. How do you deal with customers who don't understand the results that you give them?
That's a fantastic question. And what we need to do, and always bear in mind, is when we're building a work plan, is to really listen to our customers and understand what it is they actually want, what they need to solve their problem. And there is a temptation to just, you know, bombard clients with information. But actually, if that information isn't something that they can use, then we need to rethink and adapt the deliverables so that they fit with what the client's expectation is. So, you know, we try and avoid that situation by listening carefully to our clients right at the beginning of the process. We also, as I said, we don't just hand over a bundle. We present material. We're also happy to take, you know, multiple sessions if something's not clear. Communication channels are open both ways, so via email or telephone. We have project management. So the project manager will be responsible for interacting with the client and making sure they understand things, you know, the logistics of what we're doing. We also have account management, so the whole process of setting up the contract in the first place. Again, making sure that we're tailoring what we're doing to what the client needs and wants. I guess finally, it's been difficult over the last 2 years, but face-to-face meetings are also very important. So if you get the client in front of us with our computer, you know, we can make, you know, the best endeavors to explain the modeling. At the end of the day, we may be speaking to biologists or chemists. They're probably not modelers, which is why they've asked us to do things for them. So we need to explain material in those terms to our customers. And there's no point in giving material that they're not going to be able to use, unless they will then either employ a computational chemist at some point, or modeler, or employ someone else. We need to give them transferable information as well. So, you know, all the files that we generate during a project belong to the customer. So we do give them all that material as well.
Yeah, fantastic. Great answer. Good points. Shifting gears a little bit, what biological target classes and therapeutic areas do you support at Crescent?
That's an interesting question. The better question is which area don't we support? And I would say that there isn't really one. So we can pretty much do anything. You know, whether that's biologics modeling, protein modeling, small molecule modeling. Obviously, we cover that. In terms of therapeutic areas, there isn't a limit. We've worked on various things, from, you know, eye diseases, and I mentioned antifungals, bacterial anti-infectives. We've done antivirals. You know, you name it, enzymes, receptors, transporters, anything. I guess the thing to say is that the devil is always in the detail. What limits what you can do in most cases is the level of information that you have at your disposal. So, there is a range of information that, launch drugs, if you look at a launch drug, around those launch drugs, there's a trail of crystal structures and small molecules and SAR. When you're dealing with brand new targets, there might be one crystal structure, or there might be just a sequence. There may be no ligands. There might be maybe a substrate. What we do is we will look at the data stream for whatever the target of interest is, and basically we can advise on strategic ways forwards. And if that way forwards is there's just no way you can do anything with this, you don't have enough information, then we will go back to the client and say that we won't try and say we can do something that won't actually help them solve a problem at the end of the day. So some targets are just too challenging and there is not enough information, and that can be the end of the road for some projects. Actually, it's more efficient to be able to say, this is really not a goer. And maybe, you've got some other projects. Hopefully you're not a biotech that's just got one platform. Maybe there's a portfolio of projects and we can triage, there will be one project there that you can do something with. But, there's no therapeutic areas, I think, that are off limits and no targets. It's whether or not there's sufficient information to support a project.
Yeah, it's great to know that you have the flexibility, and I feel like that's also extremely important knowledge, knowing exactly what targets are worth it to go for and which aren't, because you could spend a lot of money searching for something that you just can't hope to find. So yeah, that's a really good point. All right, next question here. Martin, what business models can you offer if I would like to enter into a collaboration with you?
The main way that we interact with clients is a very straightforward and clean one. So we mainly do fees for service. So the way that works, and the reason it's very, very clean, is we just want to get paid for the work that we're doing. We don't want to hold any IP. We don't want to get any sort of stake in someone else's science. We just want to do the work, and that is the ideal scenario for outsourcing. I think when clients outsource, they don't want to give away their hard-won biology and targets, because we can help them find something. So that's the way that we normally work. In that, there are two different ways of working. One is so a very, very target-oriented or task-oriented project where it's a linear relationship, and then there are others where you can just buy blocks of time from the modelers. So we're flexible in how you deploy that fees for service work, but that's the main model that we use.
Excellent. So who is it? Who from a client organization do you typically work with?
That's a range of people. So we've been contacted by biologists or heads of medicinal chemistry. Essentially, they will be the project leads. Looking for an innovative solution to solve the problem that they have. So mainly heads of chemistry, heads of biology, sometimes heads of innovation, and again, usually we're working with a team of people. So the initial contact might be that person, but we talk to the biologists on projects, the chemists on projects, maybe crystallographers. So we can interact with any members of a team charged with doing the drug discovery project, and work through the problem collaboratively.
Excellent. So I'm hearing that it sounds like you sort of, in a way, become an extension of their team, a super specialized extension of their team to help them accomplish this goal.
Yes, essentially, yeah.
So next question here. What is the most requested type of product that you work on in Crescent Discovery?
I guess the most requested thing is probably in the hit finding part of the drug discovery process. So probably the virtual screening type activities. I guess a close second is probably SAR analysis and QSAR development, lead optimization parts of the process. But yeah.
And as a follow-up question, what services do you offer in the area of fragment drug discovery? So
we've got some excellent tools in fragment discovery. The main software tool that Crescent produced is a really excellent tool called Spark, which is a bioisosteric fragment replacement tool. Again, as I mentioned with, I think it was the third case study, because the technology is a physics-based technology, it's not limited to how big the molecule is. So we can get really nice, very accurate information on really quite small molecules or small fragments. We can describe those using the electrostatics and shape, as I've said. The tool uses that similarity metric, where we have comparing molecules with each other using basically the patterns of the electrostatic patterns around a molecule to compare them. It's a very, very nice way of finding new fragments. The tool is really good, allowing you to either do fragment growing, fragment joining, or hybridization. So that, in conjunction with another tool called electrostatic complementarity, which is a way of getting a metric on how well that fragment is fitting electrostatically into a protein pocket. Those two things together are a really nice tool set for exploring fragment discovery. And I guess fragment discovery can either be, you find fragments that bind to a protein pocket via NMR or via crystallography. The challenge is then, how do you get from there to a drug-sized molecule without losing that initial advantage of the binding of a really small, efficient molecule? So we can do that process and help fragment discovery with those really cool tools. You need more than tools as well. We've got the expertise to be able to understand and interpret the information from the fragment replacement algorithms and the electrostatic complementarity measurements in the context of protein pockets. So it's a double whammy of excellent software and an expertise that allows that to work for fragment discovery.
Yeah, great answer. So this sort of goes back to what you were talking about which compounds were practical or not. How do you consider the synthetic feasibility of a compound during the design phase?
That's a really challenging one. There are not many in silico methods that can give you good answers on that. We have, fortunately, both modelers and medicinal chemists on the team. So I'm an ex-medicinal chemist in my dim and distant past, so I can recognise when you've got a bad idea that's just never going to make it. You're not going to be able to synthesize it, or you're not going to be able to. It's not going to be stable, or it's not going to work in a binding environment. So, we can use the benefit of that experience to say, well, that's a rubbish idea. I'm going to throw that away and come up with a better idea. So it can be ad hoc, and there is no substitute for, again, expertise and experience. We do have some metrics within the tools for finding things that are more likely to be synthesizable. So the Spark case I just spoke about, there is a proxy for synthesizability, which is frequency of occurrence of that fragment in databases. So if you've got a high frequency of occurrence, it's likely that that's not a very hard fragment to put together. So those are some metrics, computational metrics that you can use to help synthetic accessibility. But mostly it's just you got to use your brain and apply that to the scenario.
Yeah, great point. Martin, can you do virtual screening?
Yes, yeah. We can do there's two flavors of virtual screening. One is ligand-centric, which is you're looking for things that are similar to each other. We also do docking as well, so you can look at what fits a protein or not. So those two flavors of virtual screening we do. We're probably the market leader in the in silico method. There are other docking algorithms out there, people that do that. But again, it's always good to have expertise alongside a method. So that's one of the benefits of if you do virtual screening. It's the interpretation of the output, and what you want to actually go and buy is the main thing. So the triaging on virtual screen is probably a more important step. Second to the actual process in the first place, what you decided to set as your virtual screening strategy can have a bearing on whether you're successful or not with the output.
Yeah, fantastic. And I think in the interest of time, we'll just have one last question here. We are interested in doing FEP calculations. Is this something that you can help with?
Yes. Yes, we've got an FEP method. We can offer FEP from within the discovery part of Crescent. It's also something that you can buy as a software item as well. This is something that's got there's a huge amount of interest in it, and we're actively developing those methods and that capability. It's already good. We're trying to make it better.
Excellent. All right. Well, Martin, thanks again. Thanks so much for the great insights, both in your presentation as well as the Q&A. It's been a real pleasure having you with us.
Thank you.
Big thanks also to the audience here for participating. And last but not least, we'd of course like to thank the sponsor, Crescent Discovery. So in closing, we hope you enjoyed this Scientist.com webinar, and we'll see you again next time. Have a great day, everyone.